Interplanetary travel has always interested people, but still little is known about the impact of cosmic radiation on human and other organisms. Since we want to return to the Moon and explore Mars, we have to think about protection of human body, but also organisms which will help us survive in long journey. Hello to all participants. My name is Ewa Borowska and I'm representing University of Warsaw with Magdalena Łabowska from Wrocław University of Science and Technology in Poland. Today we would like to present red algae as a source of efficient photosynthesis under gamma radiation during space exploration at AbGrad Conference 2021. In the picture from NASA, you can see a different type of waves and radiation, which is the most important for our existence on Earth. If we are talking about ultraviolet radiation, X-rays, and gamma waves, including ionizing radiation, we wanted to use one of the most popular gamma waves, which the source was the cobalt-60 isotope. In the picture, we would like to present interaction of ionizing radiation with matter. Ionizing radiation depends on type of passing through structural materials, such as metal makes it brittle and can weaken the material if the radiation's intensity is great enough. It can also damage electronic equipment. So we can distinguish few types of ionizing radiation. It's alpha, beta, gamma, and neutral particles, which interact less, ionize indirectly, and penetrate further. But as well as gamma radiation is a type of ionizing radiation that has an impact on cell structure like DNA, proteins, lipids, and membrane conformation. When ionizing radiation interacts with cells, the cell's DNA may become damaged. Often the body can repair a cell. However, if many are damaged or die, the body may not be able to replace the cells fast enough, causing radiation poisoning and death. Before we present our organisms, algae, which are eukaryotic organisms, we would like to show also prokaryotic organisms resistant against ionizing radiation and UV radiation. So the first one is Deinococcus radiodurans with rapid DNA repair mechanisms, which were detected in atmosphere and has the possibility to survive even after dose of 5,000 degrees and one dose 15,000 degrees. Still, some bacteria can reproduce. In parallel, human lethal doses, it's 10,000 milligrays, and for Escherichia coli bacteria, 60,000 milligrays. The second organisms are cyanobacteria from driest place on Earth and with high protective efficiency against high UV radiation. So they called Chrosidiopsis and producing specific pigments as cytonemin, which is protecting them. In Atacama Desert, unique dense matrix within the EPS, extracellular polymorphic substance, and allow for surviving the clouds in the low temperature, harsh liquids, and UV radiation. So Chrosidiopsis, the unique cyanobacteria with high UV resistance, can create the specific homes and survive inside them, still dividing. In space exploration, we can differentiate a few types of radiation. Galactic cosmic radiation created during supernovae, solar cosmic radiation primarily from solar wind and solar flares, and the radiation trapped in the Van Allen belts around Earth, and of course ultraviolet radiation. Earth's magnetic field keeps our planet safe from most ionizing radiation, including galactic cosmic radiation and solar cosmic radiation, but of course in outer space we are not protected. So we would like to present red microalgae of the order Cyanidiales occur in various extreme environments, mostly on volcanic areas such as Yellowstone Park, Atacama Desert, Cantiflegrei in Italy, New Zealand and Iceland, and of course many other types of volcanic grounds. They are characterized by low pH, high temperatures, the presence of heavy metals ions, variable salinity and high concentrations of carbon dioxide and sulfates. These organisms are the link between primary cyanobacteria and the first primitive eukaryotic cells, which were subjected to the harsh conditions that privileged on Earth during the formation of its atmosphere. What we have achieved in laboratory on these organisms, it was adapted them to high pH with the same efficiency of photosynthesis, what means that they are very unique, very highly adaptable, and with a high photosynthesis efficiency. What is more, algae cells are without cell wall. Still, they are highly resistant against of the different harsh conditions. In preliminary data, we would like to present the cells of Cyanidioschizon merola, but in the experiment, we were tested also green algae Cocomexa. But this algae has not so efficient photosynthesis, so we reject this strain. Before experiment, culture were transferred to right experimental culture flask made of polystyrene with liquid media conditions, and then transported immediately to Institute of Nuclear Chemistry and Technology in Warsaw. One place were unpacked and put in optimal growth conditions with light and temperature for activating the photosynthesis. It was around 30 minutes. Time of exposure and given doses of radiation in milligrays were calculated on the basis of gamma radiation cobalt-60 source with power 0.2353 kilograms per hour, with the last lethal dose for human 10,000 milligrams per 2.29 minutes. After experiment, axenic strain and non-axenic strain were transported again at the university and tested every 24 hours on spectroscopy UVVs in optical density 750 nanometers. And then we can assess increase of cells density. Graphs for each strains were done, and we can see the growing of algae were increasing. In further growth, they were not affected by gamma radiation. Ionizing radiation can be destructive to cells. So in specific conditions, such as space, they need to be prevented from the exterior factors by protective coatings. Gels and hydrogels are one of the solutions for growing and storing cells in a safe manner. Additives present in these structures ensure that cells can grow and proliferate, but they can also provide protection against accelerated atoms or particles from radiation, reducing their energy. One such additive could be silica, for example, which has been investigated by many scientists to reduce the impact of radiation. Many researchers are also interested in hydrogel composites as potential radiation protective coating. This material is lighter than metals and contains water in its structure, providing not only a soft protective material for immobilized cells, but also an ideal environment for their further cultivation. Immobilization of cells can be realized in several ways. One of them is the creation of silicate gels called salt gels using tetraethyl or silicate. The silicate contained in the gel can protect the cells from the destructive impact of ionizing radiation. And overall, additionally provides mechanical protection against cell damage. The second method involves blending cells with an alginate solution, then cross-linking it in a calcium carbonate solution. In this way, we are able to provide each cell with a protective alginate coating, which acquires its mechanical properties during the cross-linking process. Both methods allow the cells to grow and proliferate safely, providing them with the necessary components for survival and at the same time removing the metabolic products. For improved life support systems for astronauts, we propose methods with unique composition of hydrogel and substances which can protect algae against ionizing radiation and make them more efficient in O2 production. By using the 3D bioprinter, we can adapt hydrogel to different types of equipment and immobilize algae in a very simple way. As you can see on the picture, we immobilized algae into specific hydrogel. Further studies are also connected to regeneration of body tissue by use of secondary metabolites encapsulated into hydrogel. Our algae has the possibilities also to create biofilm on different structures, like for example cellulose. So it is also important if we would like to use other type of materials to cover them by algae. And still we are looking for collaborations. So if anybody is interested, please contact us by email. And thank you for your attention.